What's up, everyone? Welcome. We are live right now in the CannaBuzz community, and if you're watching us on YouTube, please give us a like and subscribe to the channel. We have a great episode for you today. We have Dr. Colin Bell. We're going to be talking all about root exudates and Brix and secondary metabolites, all sorts of stuff. We're going to be diving all into it, but before we get too far deep into it, I have to give a couple folks some shout outs. First off, a shout out to our CannaBuzz community. Like I said, we're live right now. Folks can join us in the chat. Every live stream we do or live interview we live stream, and folks can join us there. So if you want to be part of that group, join us by searching CannaBuzz in the App Store and download our app and join the community and use the code GROWERSLOVE for 50% off your first month. We really appreciate your support. And then also we need to give a shout out to our friend TikiMadMan. He's our big supporter of the show this year. So thank you, Tiki. Go to tikiseeds.com. He's got a sale going on right now. What was that? It was like
20% off their entire site for the summer sale. And he's still doing those combo packs, the pro pack of the Raver Runts and the Tiki Sugar. So those pro packs are still out there. Gobble them up while you can still get them.
Awesome. Well, let's get into it. Today we are excited to have Dr. Colin Bell. He's back. We've had him on before. I love talking to Colin. He's super knowledgeable about all sorts of things. And today we are going to be talking about sugars. Like I said, secondary metabolites, Brix. We're going to be getting all into it. So let's see. What else are we going to be talking about? Root zones, all sorts of things. So thank you so much for joining us today, Colin Bell.
Thanks very much for having me again. It's always a pleasure to see you guys.
Yeah, excellent, Colin. So I'm really happy to have you on because I always like to pry as much useful information out of that brain of yours that I can and pass it on to the good folks, you know. I mean, if anyone can help us elevate our game, it's typically you, my friend. So tell us what you've been up to lately and tell us a little bit about the microbial mass, folks.
Yeah. Okay. Well, thank you very much, JR. That's very gracious of you. I'm not so sure I feel the same way most days. It's a collective effort, and I learned just as much from you guys. But it's always fun engaging, and you know we've had 100 or more wonderful conversations off screen. So, hey, listen, yeah, I'm working with a great group. Microbial Mass is called MIM Horticulture. Their main products: Microbial Mass Pro. They're microbial products, and you know it suits me really well. I'm a PhD soil microbiologist, and you know what I like. Formerly started Mammoth Microbes, the co-founder, always a co-founder of any organization, and it was driven by innovation of microbes into agriculture, and that's what ultimately attracted me to work with MIM Horticulture and their line Microbial Mass. I'm at their research group. It's a Canadian group. The research group's out of Montreal, Canada area, French-speaking Canada. They are just rock stars at what they do. Cutting edge science. It was very attractive to me, and they really think out how they develop products for the market. Not only are they great scientists, but they're really engaged and well versed in cannabis cultivation in particular, which is an amazing fit for anyone that's going to be developing products for this industry. And the other group, you know, and you and I have talked about this offsite, you know, when I was looking for my next endeavor, what I was looking for is great technology, innovative group, and a very cohesive, connected team. And the MIM Horticulture group put all that together, and it was, again, really appealing. This group are shakers and movers. They are doers. There's a lot of things going on that this group is doing that's very exciting. You're going to see them continue to grow, and they're connected. You know, it's not like a group of outsiders coming in trying to figure out. A lot of these leaders in the space have been around for 20 years. And so I really love the idea of having a cohesive team that's well connected that has a synchronized vision of where they're going. Everyone's sprinting the same direction, and it's backed by cutting edge science. That's a recipe for fun, and I really like the opportunity to be helping them build out their brand.
Well, I like to see you back at what you do best, and that's getting around, learning, and creating opportunities and getting microbes on the shelves. And a little bit about their product. I noticed that when we were having our conversation, that to me it felt like the product was a kind of like a rhizo product, and it was also a phosphorus delivering product. Can you speak a little bit about its root building abilities along with its phosphorus uptake as well?
Yep. And it's a really appealing product for that way. So these are 5 different microbial strains in this product. There's Microbial Mass and Mass Pro. The more popular product is the Mass Pro. It's just a more concentrated version. That is the only difference. 2 of the microbial strains were selected for their ability to initiate root growth, root elongation at very early stages. These microbes were actually selected. They chemically signal with the plants. Pardon for the background noise. I'm in Kansas City, Missouri now, and I heard a lot of this. So I'm not so sure how to eliminate that at the moment. I'm in my studio.
It's not super loud, so it's all good.
Gotcha. Great. What growers find, and this is a Canadian-based company, and they've been in Canada for years, and through a lot of commercial facilities and all the retail shops, we see more roots with this product. Microbial mass equals more plant mass. Root mass, the other 3 strains in this product were selected for their ability to cycle phosphorus and micronutrients, so a bloom stimulant. So it took me a minute to get my head around it because I was so used to focusing on a single function, and that's how it was ingrained in my brain for a long time. But this was a very intentional formula of a 1-2 punch: roots and bloom stimulant. And it comes together actually very nicely. I'll just talk about it really quick. Some interesting characteristics. This product was tested for use 1 application every other week. And so you skip a week, and what you do is after a couple of weeks, you kind of have the roots saturated or completely colonized. And as the roots grow out, you just want to keep your confidence up by allowing the microbes to occupy the new spaces. And what happens is you can colonize a root zone, but as the plant gets bigger, the plant roots will actually grow out quicker. Then the microbes can keep up with them as they grow out. So it's nice to do those extra applications. For cloning, 1 mil per gallon. For veg and bloom, 2 mil per gallon. So it's very scalable. Super economic. This company's done a great job of actually scaling their manufacturing to get the prices where growers need them, especially in today's day and age. I was very impressed with their ability to move there. Last couple details is very interesting. They're dormant microbes. They become active when they hit the rhizosphere within a 24-hour period, which means the shelf life is way beyond 2 years. Let's just say that. So they can stay in the bottle for a very, very long time, and they'll activate once they hit the rhizosphere. And because of that, physical property, where they're in this dormant state, they create endospores. There's just high concentrations of those. They are very compatible. So you can add them in with fertility, fertigation, with a tea. They run well with all the different cleansers and disinfectants, which I love, because that's one of the messages that a lot of the microbial companies are combating right now, or trying to comply with. How do we get these growers, these sterile, clean growers, to use microbes? Because we know and believe that microbes will help improve the yield potential and the quality in those grows. But there's that disinfectant use that's pretty common these days. And so this company did a great job of testing for that and kind of bridging that gap. And so it's about positioning microbes where everyone can use them. And in a very short order, since I've been with the company this year, they've done all those things. So hats off to them. Great group.
Well, we're going to dive into our first big section today, which is all about root exudates. So to help kick us off, first off, you had a great point before we got started. First off, we should define what root exudates are. Help us, the viewers that are watching, help us understand what they are and then what role they play in the rhizosphere.
Yeah, you got it. So I think it's important to understand what a root exudate is, and we can think of those as sugars. It's carbon chains. It's simply sugars. There's organic acids associated with that also. But where it originates from is in the air. Plants assimilate all their carbon from CO2 in the air. And through a process of photosynthesis, which we've all probably heard, the plants are able to cleave that carbon from the air molecules, release the oxygen, and assimilate that into their biomass. And so I like to say carbon is the currency of all life on Earth. Organic-based organisms, even our shirts, our clothes, everything are chains of carbon with other elements bound around there to make macromolecules and bigger, very complex organisms. And that carbon makes up everything you see in the plant. Part of that carbon, though, is used to support everything else that the plant needs to take up. So although in the air, there's CO2 that the plant assimilates for that biomass and that carbon, all the other macro and micro elements need to be taken up from the soil and drawn up from the roots. And so plants have evolved to understand that they're able to nurture and take up all the other elements from the root zone by squirting exudates, root exudates, carbon, sugar into the soil, into that root zone, which it serves to feed microbes. And microbes love those simple sugars and those root exudates and those simple organic acids that the plants exude into the environment, into the soil. They grow, and what microbes do well is they cycle nutrients and they make nutrients available and readily available for the plants to take them up. And so it's simply a plant. They call it priming also, where you can actually kind of thinking about it for the folks that like to work on cars. We used to prime carburetors back in the day, way back in the day when there was just the simple old school carburetors, and you'd actually put a little gas in the carburetor to get it going, to start it. It's the same thing. The plants are shooting simple sugars into the soil, and it's revving up the microbial community so they can do what they do, produce enzymes, cycle nutrients, and create this hotbed of nutrients that plants can take advantage of so they can continue to grow and photosynthesize.
And now in sophisticated, like forested, old growth forested situations, there'll be mycelial networks where these root exudates will actually be pathways and highways for nutrient exchanges as well. Is correct?
Totally. So there's different kinds of hyphal networks, and a lot of the ones that we think about for cannabis in particular are buscular mycorrhizae, AM fungi, the Glomus species, the Rhizophagus. They're all kind of the same and name different things. And those create a network. I'll just talk about that for a second because it's something we can relate to. It's the products that we buy at the hydro shop. And that's a symbiotic relationship where those particular fungi actually infect the plant root. They grow inside the cortical cells and they create these exchange sites within the actual roots. And in that particular relationship, there's always an exchange. The plant's always giving carbon or simple sugars down to the soil, down to the microbes. And so that happens across the board. But there's different return benefits. For the case of mycorrhizal fungi or buscular mycorrhizal fungi, which we're talking about here, there's two things plants really get from that in nature and in our cannabis cultivation as well, is the fungi can grow quicker and using less resources and extend beyond the root zone or the rhizosphere zone. And so they're able to actually scavenge certain nutrients and translocate them from the soil without the plants having to spend energy growing all the way in all those different areas. And so mycorrhizal fungi in particular, they scavenge or they take up available phosphate and they exchange that in the plant root for carbon. And so that's that exchange. other versions of mycorrhizal fungi, ectomycorrhizal fungi, which are very common, JR, in the forest systems and old growth systems that grow together.
Yeah.
There's that exchange from the plant to the fungi, but that exchange for ectomycorrhizal fungi is typically carbon from the plant to the fungi, and that's why they're playing together, because the microbes and the soil microbes, whether they're bacteria or fungi, always want that carbon from the plant. But the microbes actually scavenge nitrogen for ectomycorrhizal fungi, where they find these nets, and they're just typically growing around in huge nets around the root surface area of trees and shrubs.
I think that's really interesting, because we as scientists are just starting to learn some of these mechanisms to the way these plants interact. And I think we had a point made by our previous guest, Mark, about composting, in that cannabis growers are really kind of nerding out on how to best grow this plant in a way that provides a symbiotic relationship between nature and the actual plant itself. You know what I mean? So
totally,
I think living soil is coming online indoors. You're seeing more microbial interactions, and like what you said, used to be sterile interactions. So I think all of that stuff, as we start learning, a lot of these processes are going to help maybe benefit us all and maybe feed us all in the end of the day.
I think so. There's a reason people are wanting to do it. One, it's cool. And two, there's benefits to the plants that you'll see once you start engaging in some of these practices. It's important to be practical and figure out how to scale that, where you're not doing a boatload more work to implement some of these. I think that's important, and that's probably why it's been a slow go, because some of these are just slower processes that we need to figure out for this annual crops for cannabis in particular. But I would say, hands down, if you can figure out how to implement some of these living soil practices, organic practices into your cannabis cultivation, you will markedly see a difference in the way your plant's responding, and the way your plant's photosynthesizing, and the way your plant's putting on Brix, its flavors, terpenes, aromas, all these things. It's just a very clear signal that growers get when they're able to start converting, at least at some level, over to some of these practices.
What's up, everyone? Just jumping in real quick to give a shout out to one of our sponsors, Terploc, the makers of Grove bags. You can grab a Grove bag in all sorts of sizes. They make small bags in like 8 sizes. You can even get them for your weed brand if you wanted to. And then you can get them all the way up to like giant trash bag liners. So these things can be big and small, all shapes and sizes. This is a 1/4 pound bag right here, and all you do is dry your weed, say in your grow tent or something like that. Dry the weed for about 10 days, 14 days, then stick it in a Grove bag, seal it up, and leave it be. You don't have to worry about burping or any of that stuff. You can get rid of your jars. This is going to cure it. It's going to off gas, do all the things that you need to do. All you need to do after you throw it in the bag and cure it is open it up and smoke it and enjoy it. So go to grovebags.com, use the code CannaBuzz for 7% off your next order. We really appreciate it. Thanks to Grove Bags for their support. Let's get back to the interview. So I'm going to ask this next question, and then after I do, I'm going to have to turn off my camera and go let my dog into the room, because my dog wants in. But so this next question was about how does drought and soil salinity affect all of this network and all of this interaction? People have talked about dry periods recently in the last year or so. People have been talking about dry periods, and I just kind of wonder how all of that kind of impacts this network that I'm trying to build up.
Thanks for the question. I'll let you go get the dog and I'll start answering it. Yeah, there you go. Well, listen, there's everything we do when we're growing a plant is interactive. And I think it's very important to understand that, going back to the concept of photosynthesis. You can't photosynthesize without the sun or without some type of light source, but it requires more than that. It also requires water. And so as soon as you start changing the dynamic, the hydrology, the water availability, you're immediately influencing the plant's ability to photosynthesize. And so that's a huge part of that interaction just alone. The turgor pressure, there's a lot of things, the flow of nutrients, the ability of the plant to translocate, everything happens through that water column within the plant. And so to be able to do that requires water. And also, you think about the photosynthesis and requiring water. If a plant cannot photosynthesize, it's not going to be able to build up enough of the solutes, enough of the solid materials or the soluble material, and the sugar content to allocate its resources towards defending itself against the environment, towards creating, we talked about secondary metabolites and all these other defense mechanisms. And in some cases, secondary metabolites are something that we really like in the later stages of the plant growth for cannabis. Secondary metabolites, terpene. And so there's some of these that we want the plant to be able to turn on and regulate. And all that happens with the ability to maintain adequate water. And so with salinity also, I would say that there's a lot of things that affect the plant during salinity or these high salt concentrations. A lot of that actually turns out to be, at least in natural systems or soil systems or living soil systems, the inhibiting the microbial abilities over time to function appropriately and actually cycle and release nutrients that the plants need. Again, it all ties back to photosynthesis. So I think, thinking about the process of photosynthesis and everything that plant needs to photosynthesize is exactly all these different pieces is exactly what we want to optimize. The plant should be photosynthesizing optimally. And without doing that, it's not creating photosynthesis. Without the photosynthesis, it's not maximizing its Brix and soluble content. It's not being able to allocate those resources for growth, for defending itself, for maximizing all these properties and phenotypic potential, or for feeding the microbes in the rhizosphere zone, which ultimately feed back. We don't think about this enough. A lot of the photosynthesis is dependent in many cases on some of those soil properties. And we're talking about, at least in soils, a lot of these micronutrients and cofactors, trace metals that are important to optimize the regulatory processes and enzymes in photosynthesis. And so a lot of these trace metals can be highly bound in the root zone, and the plants can't take them up, but microbes do a great job of doing natural chelating and mobilizing these trace elements, these trace nutrients, not only micronutrients, but trace metals, which the plants have to optimize photosynthesis.
Especially like in flowering, like when you need large amounts of magnesium and sulfur to finish off your flowers in a very aromatic and terpene-rich way. A lot of times people talk about root structures. Some people say that, the bigger the roots, the bigger the fruits. Can you talk about kind of the architecture of structural roots in a relationship to building strong root exudates and relationships?
Well, it's not my area of expertise, the architecture of roots. I think plants have a natural tendency. What I would say is early on, plants want to focus. I would kind of phrase that question or phrase my answer to talk about the focus of energy for plant growth. We want the plants to focus on that root growth early on. Kind of evolutionarily speaking, roots do a couple things. The first thing they do is they anchor the plant. And sometimes they need some type of substrate to anchor into, to grow into, just to hold upright. And so one of the main functions of roots is actually an anchoring property. And so roots are nurtured or tailored by the plant to create that support. And there's some actual physical properties that the plants get triggered by to grow those roots appropriately so they can hold themselves up. Plants are very responsive to different physical stimuli and light stimuli. They will grow where they need to. And if there's a bending or a physical property that they feel, they'll compensate pretty quickly for it. And early on, they can do that through root growth. Ultimately, the plants, especially annuals, want to slow down a little bit in their root growth and focus on above ground growth. And so I would say at some point, the plant probably senses where it has enough energy allocated into the root zone where it's actually taking up enough nutrients and enough water within that particular zone to grow upwards.
Oh, that makes sense.
So, yeah. Sorry, go ahead.
So I have a question about that actually. So, especially on the point about growing upwards, because I've got, several plants outside right now that are growing in various states of flower. Some are like very far along, some aren't very far along. And I've noticed as long as I just keep top dressing with more like worm castings and, I keep it covered with a mulch layer, eventually those roots are going to be like the whole thing is just going to be a root bed, basically, if you have that mulch layer. Even if I'm like well into flower, 5 weeks into flower or whatever, is the plant still eating up all that nutrients and I'm still helping it out, right? Or is it just kind of, I'm just curious, what do you think it's doing at that point?
Well, those roots are functioning 100% to take up. There's a joke that we used to make in graduate school. We were microbiologists and we were soil guys, and we used to always have fights with the plant people, although we worked together in all of our studies. We did plant microbe interactions, and so they did the plant stuff and we did the microbe stuff. And then we put our data sets together to tell an ecosystem level picture. And so it's wonderful studies and wonderful groups. But the truth is, physically, plants do one thing more than anything else. They're experts in it. You want to know what it is?
Grow.
Getting me high.
They do those things well. They do them very well. And this is a little bit of a snarky joke. Don't take it that way, but because it's true. They suck. They suck CO2 from the air. They draw or suck water and soluble nutrients from the soil. That's all that plant's trying to do, is draw in from above and from below. That's all it's doing all the time. That's all it wants to do.
It's just totally sucking, and so that it can make the next generation of suckers.
That's exactly right. So it can make baby suckers that will grow up to be adult. Yeah.
Suckers.
Listen, that's what they do. And there's a balance, if the plant above ground is getting enough from the below ground, it's not going to necessarily feel like, because energy and resources are precious. When you're anything other than a human, everything is valuable. We're very wasteful as humans, but as a plant, I don't want to send anything to the root zone. I don't have to, because I'm obligated, I'm beholden to success. And success for a plant is spreading my genome to the next generation for all biology. And I can't do that by focusing just on roots. I need to do that by focusing on my above ground canopy, my growth, and ultimately going to seed. We trick the plant, and we don't necessarily always allow that, but that's the plant's inspiration.
Right.
And so when the plant would sense, hey, my root zone is sufficient, I feel water coming up all the time, I hydrated, I'm getting my nutrients out, it doesn't need that. Whenever there are limitations, I would imagine more growth will be regulated, and there's always a little growth, but it slows down substantially towards the later stages because the plant only wants to focus up. And what we find, and you'll see this, and it's very interesting, even in a hydroponic setting, let's just talk about that for a minute, in a rock wall where you can actually see the roots.
Yeah. And
they're not that big, but they're getting fed. It's just an anchor, and they're getting IV fed all the time, where the plant's tricked thinking, hey, I don't really need to put a lot of resources in my roots. I'm holding myself up. So physically, I'm
right
in control, and I'm giving myself all the hydration and nutrients I need because it's being fed. Well, if that was in a different soil environment, the plant might not have that much nutrients, or a different hydration regime where it would need to extend its roots out more to suffice the demand from above ground.
Mm-hmm. Very cool.
And now one of the things that people always throw out there is that if you're using a lot of microbes, you don't have to pH. Can you speak to that a little bit?
Well, I think it just depends on the environment you're talking about. I mean, what you don't want to do is risk shocking your plant with a very drastic range of pHs. I don't think that's fair. So I wouldn't recommend, if you have even if you have a nice soil environment and you have a lot of microbes, yes, it's going to be very buffered. I wouldn't worry about it. But if I had a nutrient solution that I'm adding regularly to that, just as a hypothetical example, and that nutrient solution was very acidic, then the plants aren't going to like it. And ideally, will the plants bounce back from it, and will the microbial community in a soil bath be much more buffered than, let's just say, in a rock wool or a hydroponic environment? Absolutely, positively. But there's still this range of, I would hesitate to say that it wouldn't do anything, or it would be ideal, because it wouldn't. pH is the single most influential factor to influence microbial diversity across the globe. It's been well shown. And so you're going to impact life by drastically changing pH, even for a short term. And I guess how I think about it, and I'll shut up about it, but this is an annual crop. We don't want to shock the plant ever. We only have so many hours in a day and so many days before we're yielding. And I would say a pH shift can put the plant, even if you don't see it visually, it can put the plant, can pause the plant's optimal uptake for half a day. If you're in systems, you can have pH shifts. I've seen them in grows where it happened 5 days earlier. The plant didn't show it for 5 days when they were actually looking with their eyes, and that's when they realized there was a problem, when the plant actually showed an effect 4 or 5 days later. Then they had to reverse that, bring the plant back, and it took 10 days from the day that the pH got messed up in that one tank before the plant started wanting to drink up again. So you lost 10 days of valuable annual crop cycle in that particular crop for that table. And it was a horrible turnout. So living soil, the microbes help mitigate that a ton.
Yeah.
I would still, not want to dump a pH of 3 in my soil. You're going to get some type of adverse effect that will turn around quicker in a living soil or in a microbial soil than it would, obviously, in a rock wool.
The other question is if I'm in, like, say, a cocoa peat compost kind of blend, which a lot of soils are in a bag you get nowadays, and I'm wanting a fungally dominant rhizosphere along with a bacterial dominant rhizosphere, what kind of pH number are my ranges that I'm kind of shooting for?
I think you just need to keep with the same range that the plant likes. I mean, at the end of the day, you're focused on plants. You're not focused on the microbes. And the plants are going to feed those microbes what they need to be. And so, that's where the pH of near 6, I think, is optimal. The bacteria will do just fine. The fungi love it just fine. And so it's a very nice range. When you start going too acidic, you'll knock out the bacteria. Bacteria hate really acidic conditions, but life in general hates really basic or it gets challenged with extremes on basic or very acidic conditions. But there's a nice range where fungi can persist, and many bacteria can also.
I recommend 6.0 to 6.2 for just about anybody.
Yeah. Well, I would say that there's a range of biology. How I think about it, biology typically has a range of at least a full unit. And so a microbe between 6 and 7. If microbe like 6, typically a 5 to 7 range, will be fine for it. It's when you go outside those ranges that it's going to start getting very distressed. And when you get near those endpoints, it's going to start maybe affecting the way it acts, but it's not going to completely deprive it of function.
So let's get into our next big section here, which was all about Brix. So what is Brix? What is it? And what does that refer to in the context of cannabis plants?
Yeah. Well, Brix is a pretty interesting thing for cannabis plants. And there are some people that have pretty strong opinions about it. I don't know that many people that are actually measuring Brix in their cannabis grow. And so I have my own opinions about it, but what it is, it's called Brix. It's spelled Brix. It's traditionally a sugar content. And you can measure it you can actually measure Brix in roots. You can measure it in stems. You can measure it in leaves. You can measure it in a fruit or a flower. And they're typically going to have these different characteristics depending on the area of the plant. It's most commonly measured in the leaf. And so it's a percentile or a unit of sugar relative to a certain amount of a solution that you can extract from the plant. So a certain amount of water. And it's usually referred to as I've heard it referred to as terms of glucose content or sucrose content. But basically, if we just think about it as sugar content, it's 1% would be 1 unit. And the units typically range, or I guess what they say is for most crops, an optimal point for Brix would be 12. So the unit of 12 for plant Brix would be optimal for a couple different reasons, but it's very, very hard to achieve. So just to be clear, Brix is the amount of sugar in the solution. It's all the solutes, but mostly that consists of sugar. You'll read a lot of things about Brix where it's all the different ions, including sugar, but it's a very, very small portion of all the other ions. So you can consider it as a function of sugar.
So how do I influence my Brix then? How do I get them to the right number? How do I get them up?
Yeah, so that's what's weird for me, because it's a byproduct of how you're growing and how healthy your plant is. So Brix is thought of as a health metric for your plant. And it comes down to the photosynthate. And I use that word photosynthate as just the carbon or the sugars that your plant is fixing from the air, from CO2, using photosynthesis and assimilating into the plant. So things that affect Brix are nutrient, are water, are the ability for the plants to photosynthesize, all these details. And I'm not so sure you need to think much about Brix, but there are some interesting details around Brix that I think are worth bringing up. So it's been a point of, I don't know, the topic kind of ebbs and flows. Is Brix important? And I think the answer is it just depends on how focused you are or the questions you're trying to answer in your particular grow. So they say that an optimal level of Brix will help your plants resist pest pressure. So that's one of the things that I've heard quite a bit. And I did some research. I don't think about Brix in cannabis a lot. You think about it in tomatoes and you think about it in grapes and some other crops, and it's sugar in it. Extra Brix means it's extra sweet. I don't really think about cannabis as being a super sweet crop, but I was thinking about and doing some research for this particular talk or this particular show. And a couple of folks have suggested that it's very hard to have a very high Brix content if you're in a completely hydroponic environment. Where you go to more of an organic environment, and organic growers typically have higher levels of Brix. I thought,
and
okay,
Brix,
interesting.
I thought
and
that was
Brix are responsible for what I think we should maybe go ahead and get ready to jump into is those secondary metabolites. So can you talk a little bit about how the Brix relate to flavors and aromas?
Yeah, well, and I'm going to relate this all to pest pressure also, because it all comes down to sugar resources. So there's a concentration of sugar or carbon in the plant, which it can use to assimilate and to create different properties. It can assimilate into biomass. It can assimilate into root exudates, which it can actually feed the microbes, which in turn regulate and help promote photosynthesis. It can create things like terpenes and a lot of other secondary metabolites that we think about for not only the quality of our plant, but also some antimicrobial properties. I was doing some research and I was listening to a guy that had done a lot of work on Brix, and he was actually geeking out on the insect response to different Brix contents, which I thought was fascinating. And I'll be sure and share the name of this particular researcher. He was an independent kind of a professional researcher, and he had his own company. He was an entomologist, had a PhD and an undergraduate and a master's degree studying insects. And a lot of what he did was study olfactory responses, smell responses between plants and insects. And he did a ton of work, but he did a lot of studies on looking at very meticulous Brix levels and how different insects would infect a plant at these different Brix levels. And it was actually pretty fascinating. I took a note because I didn't memorize everything he had to say, but I want to bring it up. He brought up that certain groups of insects would be attracted at different stages of a Brix level across the plant growth cycle, depending on their preferences and their susceptibility to sugar and how they're able to eat on the plant. So, for example, aphids are a very specific group of insects, and they're these suckers that actually have to stick their feeding tubes into the plant, and they stick that and penetrate the plant, and they actually take up the labile substance, the internal, material, the Brix, and solute within the plant. The interesting thing is higher Brix contents for aphids, the plant cuticle cells are harder, and so it's harder for the aphids to penetrate and actually feed off of those particular plants. And we're talking about leaves in particular. And secondly, that group of insects is a lot more susceptible to higher levels of sugar. And so they can't actually drink higher levels of sugar. And so they'll typically attack a plant when its Brix are closer to 7.
Yeah.
And so we have these other groups that can either have or more capable of breaking down plant material. The chewers is what they called them. He had different groups of suckers and different groups of chewers as far as these insect classifications. And so the more competent you are at chewing up plant material, taking up more sugar, you would attack the plant at higher levels of Brix. And so the Brix physically can help withstand insect pressure from the plant. They also, the plant also can develop these different secondary metabolites with higher Brix to help repel or even in some cases kill insects and microbes in those cases as well, with higher Brix levels.
That's so you had me I have kind of like, maybe it's a dumb question, but and it's also just kind of coming from observation because I've been hanging out a lot in my garden. There's a picture behind me of actually a praying mantis that I took. But I'm looking at my plants that are in flower, and I'm noticing that the plants that are like deeper in flower, they have bugs stuck like in the flowers, like little little tiny bugs. Sometimes it's like a ladybug, and I don't know if like the ladybug got knocked out by the terpenes or whatever. But the question, kind of explain it to me like I'm five, question was almost like, it's just kind of mind blowing to me that the plants evolved these different terpenes, these different smells and aromas and tastes that we love, and we love to explore. It evolved these as defenses, and that there's probably like some sort of bugs or other things that it's repelling against it. And it's just fascinating to me that that's what's actually going on there.
I actually posted a post today. There's a researcher up in British Columbia, Canada, and he was talking about the terpene productions in a pine forest will actually change weather systems. The molecules of the intense pinene terpene will float up into the ozone, attach to other molecules that will weight moisture over, and then actually start forming moisture for a droughted out pine forest.
How cool is that? That's
incredible. How cool is that? And that's another example of how I think that as scientists, we're finally being able to wrap our mind around some of these functions that happen in nature that are ultimately going to be the massive benefit to all of us. Especially as things like CRISPR technology start becoming more mainstream and stuff like that.
No, I was going to make a joke that it's almost science fiction, but what it is we're just learning that nature is pretty freaking powerful, and I just love I was a part of making a product, a fungicide, a miticide, and insecticide product not that long ago. It was years, and I learned a lot during that process. But what we knew was there's natural chemistry. You look out in nature, you look out in your backyard, those plants aren't all dying from insect infestations, from fungal infestations. When I've been driving all through Missouri and everywhere else, there's natural systems that defend themselves. Well, how do they do that? Through natural chemistry. And thyme was one example where we learned there's some, and a lot of herbs in particular have very interesting chemical properties in alcohol groups, rosemary. You see all these. Well, there's a reason. There's chemical constituents that these make, and at right concentrations, they'll just drop insects and fungi, and they'll disrupt their cell membranes. So at that level of the insect or the microbe, these things are deadly. And if they and they're not necessarily always having to allocate. We talked about that, the choice of allocating resources towards a particular need or function. But the plants, someone asked me the other day if I thought plants were conscious. Do plants have a consciousness? And I thought about it. And I thought about it for a sec. I was like, well, I would say yeah. And that's how I explained it. If we can define consciousness as an awareness of our environment and an ability to functionally respond, and in this case for survival or defense or whatever that is, I would call that a clear consciousness. It's nothing that we don't do every day as humans, and plants do that on a regular basis. And so talking that through with him, we were like, yeah, we both agree plants are absolutely positively conscious, because of some of these reasons that we talk about. Not only do they grow and respond to different weather climates and wind and sun and everything else, but they respond to the environment and other organisms around them.
Absolutely. And I would say that in my lifetime, I've been accused of being a tree hugger, but go find a big tree, give it a hug, and tell me you don't feel something. Tell me you don't feel something, right?
Totally. Yeah.
I mean, there's something majestic about something that's been standing around for over 200 years. They obviously know things that I don't know, and they can't even move. They have to stand in one place forever.
Well, you mentioned something about those fungal and those microbial networks that connects those forests. And so there's a lot going on that the humans are just now starting to accept as reality. And the science is uncovering all these wonderful associations and networks and communication highways that nature facilitates. It's absolutely amazing.
So I guess the natural question I feel like as a grower or someone who's always trying to make their stuff better is, how can I make my plant have more secondary metabolites? Like I asked earlier, how do I get the numbers up? But and I also had a kind of a second follow-up question, I guess, that you had me thinking about earlier, because I think you made some sort of comment about the plants and sugars, and you just made me think about being at the grocery store and noticing all the sugar additives or whatever,
Yeah.
and people, and I just, a lot of people add sugars like in flour because it brings up the terpiness or whatever. Like, what's going on there? I guess because that's kind of what we're talking about, right? The secondary things, like, and are these expensive bottles that people are buying and dumping on, are those doing much, I guess?
Are you talking about the carbohydrates or those sweeteners?
Maybe that's what it is. Yeah, sorry, I'm throwing a lot of questions at you, but yeah.
So you did throw a couple questions out there, but thinking about this, and I was thinking about this a lot, and I still don't understand, but I think the answer is just pretty simple. That the sugars that we add into the soil, the plants have a hard time taking those up. Plants take up very small molecules, extremely small molecules.
Ionic or enzymatic, right?
Yeah, well, enzymes, microbes create enzymes, and plants do some as well. Microbes do much more, that break large chained molecules into small chain molecules, and that's where you're making nutrients bioavailable, where you're cleaving off nitrogens and phosphates from larger chains the plants can't take up because it's size limiting. It's just like a filter. Those roots are like a filter. They have a very, very small hole and ion exchange pumps that they can pull things through, but they can't pull a chain. And so microbes break those down in small sizes so the plants can take them up.
So if you had a preferred sugar to boost your microbes or supplement your microbes, at the end of the day, cost effectively, what's the sugar you would recommend the most?
I don't know what the costs are. I've heard a couple different things, but I think simple sugars work very well. I think I would say that a diverse substrate or a diverse carbohydrate would be my preference, because and just to get to the point, sugars are feeding the microbes, and I think more and more that's the common consensus. The microbes are doing a lot of the work and cycling the nutrients and liberating a lot of these things that a general base or synthetic program is not delivering to the plants. But the microbes can close that gap, and if you nurture that microbial community, they're actually delivering all those other components that that the plants need. And so that is triggering these terpene productions for a couple of different reasons. One, it's allowing, think about the microbial population, and JR, I'll definitely answer that question too, but the microbes are delivering all these constituents that ultimately stimulate photosynthesis through the not only the main macro nutrients that plants need to facilitate photosynthesis, but through the trace metals and the micronutrients that the plants also need to maximize or optimize photosynthesis. Photosynthesis creates photosynthate. Photosynthate maximizes, along with water availability, the plant's ability to put on the Brix. The Brix allow for to maximize the plant's ability to allocate resources towards towards regulating different properties, including secondary metabolites, which is terpenes. And the microbes do one more thing that's pretty interesting. We talk about plants being aware, being intuitive of their environment. Well, plants sense those microbial relationships. Plants can sense fungi. Plants can sense bacteria. They're engaging at this level we can talk about, but we can barely conceive. We know these are happening. We can measure some of these things.
Is that what gives the leaf boners? Because I feel like whenever my soil is well inoculated, my leaves are just like boing. Is it from that or hydration?
Well, I think it's probably a little bit of both, but it's definitely your plant's ability to take up as much as it possibly can. So there's a little bit of turgor pressure that's occurring there. And so they're just really happy and they're rigid. And that's what happens when they're praying like that a little bit, is what some people call. But ultimately the interaction allows the plant to realize, hey, man, I got a lot of stuff here that I'm taking up, so it can allocate. There's also a little bit of immunoresponse that we talk about. If the plant signals something, it's going to allocate, in some cases, secondary metabolites because it has the resources and it feels like it needs to. Well, microbes, even the beneficial microbes, will trigger some of that. The plants are there. They're like, man, I'm sensing a lot of microbes. I better allocate some of my resources towards secondary metabolites to make sure that I'm in a defensive mode, which in our case, we love that. It's safe and healthy for the plant, and it's ideal for the farmer to want that. And so plants can allocate these resources towards more resin production, towards more terpene production as it's growing. So I think more and more the feedback of even the Brix and allocating all those resources comes from the root zone. We know that, but comes from the microbial associations in those root zones specifically. So it is full circle. You can't really have one without the other. We know that if we have a very healthy microbial organic environment where there's a lot of resources that are being liberated for the plant to take up, it's going to express differently than if we have a synthetic, non-microbial or limited microbial environment. I've seen it a bunch, and it's neither here nor there. You know, people like to grow all sorts of ways. I'm just saying that I believe the plant's happiest and it has more potential from a photosynthesis point of view. It requires all these other details as well: environment, CO2, light, water, all these things. Microbes are that last piece that are just going to allow the plant to push photosynthesis to that next level.
There's my dogs. I was just gonna say we're coming up on the last few minutes of the show here. So JR, I think you're gonna have to kick us off with the last few questions or so.
Yeah. Excellent. So I mean, obviously we've done a lot of talk about Brix. And Brix are kind of a hard thing to measure. So at the end of the day, are we trying to measure our Brix? Are we just using our Jedi skills to know that things are functioning the way they should function?
Well, it can be cumbersome to measure Brix. And so you would want to have a specific reason or objective for doing it, in my humble opinion. But there could be some reasons to do that. First of all, if you don't know how to measure Brix, you can get a pretty simple handheld refractometer. And there's a very simple process. And let's just talk about leaf Brix. I think that's a functional way to look at it. You can pull leaves off of a plant. You can actually shove them in a garlic press and press them and get liquid. And a refractometer, maybe you can show a picture or something. You just drop a couple drops of the leaf, squeeze through the garlic press, the leaf juice, and then it's just a measure, and it refracts light and bends light around, and it's calibrated to measure the concentration of sugar or Brix in that particular solution. So at the end of the day, a way to or a purpose maybe to measure Brix would be optimizing or asking the question, how can I optimize my growth, my inputs? And so I was thinking about an experimental design. Obviously, it all requires time, and time is money. But if you really wanted to optimize or have a question of, if I add this extra input, do I think you can ask the question or hypothesize, will this help my plants photosynthesize better? And let's just focus on photosynthesis, because that's what Brix is. At the end of the day, that photosynthate. We want our plants to be working as much as they possibly can to photosynthesize. And so a refractometer, it's not a perfect tool. It's a little bit of a general tool, but it would be relative across different plants. If you're using that for a bunch of plants in a day, you can get a relative measure of how your plants are performing. And if you wanted to do an experiment where you're adding a couple things, thinking or adding something to your grow, hoping that it's going to improve your plant's health and when we're talking health, your plant's ability to photosynthesize more, to work more in a nice, natural, healthy state. You can measure photosynthesis across a week of adding that extra whatever to your grow, to your practice, whatever that is. And if you could see the Brix level through the refractometer, that measure increase across the course of a 24-hour period, a 48-hour period, or even longer by using that additive, I think it would be cool. Like if I had this one thing that I added in and I noticed photosynthate or Brix increasing, and you can typically measure, you can start to measure changes after about a 24-hour period. If you could see those shifts of half a Brix or a full Brix unit over the course of days, you might be onto something. And so your environment and everything you're doing is constrained in your grow. And so you're not going to necessarily change the dynamics of your Brix, but if you wanted to test if something that you're adding to your grow would actually significantly improve that characteristic of your plant, I think it would be a good way to try to measure that. Did that make sense?
JR, you're on mute. We're gonna have to get into our last few questions though. So JR, don't know if you had a follow up there.
Well, I was just going to comment on the fact that, those secondary metabolites, the terpenes and the flavonoids, those are all things that are something that our human senses can pick up on. And like Colin's saying, if you do have an additive that you are spending a lot of money on, you want to be able to quantify that that's actually bringing something to the table. Mm-hmm. And I think, whether it's measuring Brix or just sitting down with your bioassay and taking a bong hit, it's all important to understanding, And so part of my final shout outs, I've been doing these fun questions at the very end, Colin. So and you have been a world traveler in the industry of cannabis.
Yeah.
What has been one of the greatest examples of growers' love that you've experienced in your many travels?
There's a bunch. I'll share one in particular. I was in the most isolated big city in the world. You know what it is?
Yeah.
Perth, Australia.
Oh,
it's on the very far west coast of Australia. Yeah. Australia is basically the size of the United States geographically.
I didn't realize that until recently, by the way.
Yeah, so and about 1/10 the population, so it's incredible. And going to Australia is like going back in time in the cannabis industry in the U.S. 15 years ago or more, where everything's still under cover, under wraps. The stores are still the hotbeds, and everyone's coming for information, and so it's under wraps. But you get a little more confidence when you're talking with people in the store. And I remember being in a store and, giving a spiel, talking to micros. I've seen the world one grow shop at a time. I've been around the world four times, one grow shop at a time. I've been to thousands of grow shops. And so, you go, you engage, it's cool. There's so many more similarities and differences. They say this industry is kind. And just that saying, I think that everyone here in this industry is unified around a plant, and it's a beautiful thing to be. What I would say is, I was talking to this one guy, and he's slowly, very, very standoffish, and I gained his confidence and his trust, and we just started talking shop, and he was so psyched about getting some samples and trying some of these products that I had, and it was really fun. And you saw him just getting more open. And finally, it's like that first time, you remember way back when you could actually talk to someone. You could tell this guy was alone, and he was alone for the last 15 years, and he had no one to talk to. And he's taking back routes on the way home, and he lived 45 minutes from the shop and all this stuff. And he was like, mate, I got to show you this product that I've been growing. And he was just so psyched, and he was dying to share with someone. And I had a long day ahead of me. He's like, can you please wait? I want to give you some. I want you to, and he was so proud of it. And you could tell the passion. And I was like, sure, I'm waiting for you to go do it. So he drove all the way home, drove all the way back. It was about 2 hours later. I just hung out at the shop, talked to people, had fun just for that. And it, it wasn't just that act, but you could tell what it meant to him. And today we were still going back and forth. He was a very shy guy, very discreet. But you could tell it was meaningful. Like he was like, man, having to be able to connect and just to share his passion with another human. That's something that most people in this industry went through, where you really, if you wanted to be safe, you kept to yourself. Yeah. And maybe you had a close friend or maybe a close, very discreet network. But just seeing that, and time again, that's just what I saw is, people for 1 minute were able to talk to someone that they could trust and engage with and share some of their, some of their hard work and their efforts. And I ended up on all these trips, most of them through the UK and Spain. Everyone would want to give you some of what they were growing. Especially if it's some stuff that they're growing with your stuff. And, it was pretty kind of, not necessarily legit to have bags of weed. Yeah. And I'd be carrying around these big sandwich bags full of samples all through the UK. I remember once I was on a train and I was smelling something. I was like, uh-oh, someone has something. That I was looking up. I was like, oh man, that's my stuff. And so I got off the train and figured out how to do something different with it. But it was just, navigating in a space where people were navigating and taking risk and bringing you into their life because you're trying to help them. And they were really proud of what they were doing. And so there's just this thing that I'll never forget. All the people I met around the world that honestly could have been in the same network. And it felt like this one same unified network of people all over the world that had the same passion, that had, again, much more similarities than differences. And I was just amazed by it. It was a really cool experience.
Well, I'm hoping that love of the plant and the love of sharing. I mean, I think that's one thing that's very common in our community is our desire to share what we've created and to see other people enjoy it in the way that we've enjoyed it. And I think that love for our plant can help build love for ourselves. And if we can keep from being separated from the love we have within ourselves, then we're all going to be able to grow like the plant, and we're all going to be able to produce and shine. And I thank you a lot, Colin, for all your generosity and all of your kindness over the years. When I first met you, I was just a fanboy coming up trying to get a sample, and we've developed a relationship over the years, and you've always been in my corner, and I can't thank you enough for that, Colin. I really appreciate you.
Hey, back at you, JR. Our friendship goes deep, and I've always appreciated you. I remember the first time we met, and I really cherish our friendship, and always there for you guys.
Well, thank you so much, Colin. How can people keep in touch with you, keep up with your adventures, shout out your websites, whatever it is?
Yeah, cool. So, I have the same Instagram, Colin W. Bell, the real Dr. Colin. So that's there. And so you can always hit me up there. The Microbial Mass Mim Horticulture, it's mimhorticulture.com. At Mim Horticulture on Instagram. You can hit them up. Really great group. There's Jeff there's Frank at Mim Horticulture. Very, very cool guys. They're based in Canada. And again, they're just rock stars. And we're all here doing the same thing. We're all here trying to focus on, the plant and allowing farmers to be more successful and enjoying the science of growing and inventing and just the whole journey that we've been on together. It continues.
It's been crazy. It really has been crazy.
It's been pretty crazy. Yeah.
Been crazy, man.
Yeah. It continues. It's ever changing. We are guaranteed one thing on earth, and that's change. We're guaranteed change, and so we might as well embrace it and see where it takes us. It has been one wacky ride.
Yeah, it has.
Yeah.
Well, thank you so much, Colin. We really appreciate it. We had a great time hanging out with you this evening. JR, thanks for putting together a great selection of questions. We were able to cover pretty much everything that we wanted to cover, so really appreciate your time this evening. I hope everyone has a great week. Thank you so much for watching the show. Like I mentioned earlier, make sure to like the video, subscribe to the channel, keep in touch with what Colin's up to. You could actually look up our other videos with Colin. We have at least one other interview that you could look up with Colin, and all of our other interviews on the channel. Also, shout outs to our friends over at Grove Bags at grovebags.com and Tiki at tikiseeds.com and TikiMadMan.com. And as always, JR, we love to sign off the show every week, with our slogan, which is Growers Love, everyone.